Alright. Shitsumo. Questions? Any questions? Any questions? Anything that's not clear or would like to have some information or anything else? So as I said, I'm not going to see anything new today. So it's only if you have any questions, then I can respond to it. Otherwise, that's it. That's it. So if you have any question, don't hesitate. So, don't hesitate. If you're all silent, I'm just going to leave in 5 minutes. So don't hesitate, it's your chance. If there is anything you don't understand, now is your only chance. Next week it's too late. Question. Question, I want questions. I want questions.
I have not read the book.
Oh, so for the test, can we bring the book or no? Because then, different type of preparing.
No. Dictionary, no problem. Dictionary, no problem, but not the book. Not the book. I don't think we have seen too many new things, so that's manageable. That's manageable. Okay, so no book. No book.
Can I use dictionary on my phone, or does it need to be the paper one?
No, you can only bring 1760 old dictionary, not electronic, vellum paper and everything. No, so yeah, the problem is nowadays mobile phone, you can actually check whatever you want. So I kind of want to trust him, but usually mobile phone is actually not a good idea at exams. So.
We need a calculator.
We
need a calculator.
Calculator if you want. I don't think you need it, but you can bring it if you want.
I don't have some.
But I don't think it's needed. I don't think you need to calculate things that require a calculator. I don't think so.
Does the test mostly focus on stuff after the test, like the previous test?
Come again?
Is this exam focused mainly on stuff after the previous test?
I guess probably
Like a
3/4, at least a half of the questions will be of the new things that were not covered by the previous test.
5, 6, 7.
Yeah, so at least half would be 5, 6. Yeah. So. And maybe the chapter 1, very little things, very little questions probably. So, yeah.
Quite a bit of stuff to memorize.
That's it. That's it. どうですか questions 質問どうぞ日本語ででも OK
Last week,
Mhm.
we saw a protein which attracts RNA polymerase 2, and the protein
Which in bacteria or in eukaryotes?
eukaryotes. This protein can recognize the direction of direction that RNA polymerase 2 moves
Yes,
to.
yeah. Well, it can recognize. No, it's not exactly true. It does not recognize the direction in which the RNA polymerase move. It recognizes the direction of the gene.
Of gene, yeah.
And then because it recognizes the direction of the gene, it recruits the RNA polymerase in the right direction.
How does the protein recognize the gene? If it were guide RNA, it can be possible, but how protein can precisely attach to DNA?
Okay, so first of all, there are a lot of different things in your question. So, what do you mean guide RNA? The term guide RNA is something that is specific to the Cas9 nuclease.
Yes,
Okay.
I hear that only in Cas9.
Yes. So then so in case of Cas9, you understand how it recognizes.
Yeah.
But the general transcription factors or the sigma factor in bacteria, they also recognize the direction of the DNA. The DNA is a double helix. What do you think? How can direction be recognized? I can't tell you, but if you think about it, I'm sure you can understand. So we are talking about double-stranded DNA, right? Before transcription occurs, before the two strands are separated to allow RNA polymerase to synthesize RNA, the two strands will be together. So we have a double helix of DNA, but in only one direction is a gene. The other direction, maybe there is nothing. So how can a protein actually detect? How can a protein detect which direction to go?
Maybe protein, some structure of protein can selectively attach to
Okay, let's go.
base.
It's for everybody. Everybody can answer. So, what is the most basic information that DNA gives? For all the protein that bind DNA, what is the most basic thing that is recognized by all these proteins? You see the double helix, and what is the most basic thing that protein will recognize that are different from one part of DNA to the other part of DNA? What is the most basic thing?
I think it's base.
What base? What do you mean base?
A, T, and G, C,
So
and
that's
purine
the sequence. That is the sequence.
sequence.
The sequence of DNA is the most basic information. Okay, A, C, T, G, T, G, A, whatever. So, that's the most basic thing. So if we draw schematically, well, we have a strand of DNA here, and we have A, T, C, G, G, C, T, G, A, whatever, and here we have T, A, G, C, C, G, A, C, T. Okay? So, if, let's say, this sequence GGCTGA indicates the start of a gene, then there is a protein that could bind to this sequence and detect the GGCTGA and know, okay, so this is the start of the gene, so the protein will bind in the right orientation, okay? Because there is a specific sequence at that location. However, there is something a little bit confusing, is that in this case, we are talking about a double helix here. We are talking about a double helix, not about a single strand. Okay? Not about a single strand with the G and the C, the base sticking out and available for binding. In this case, it's a double helix. So these things are making bonds with each other. 2, 3, 3, 3, 3, 2, 3, 2. So the base pairing, they are not available for protein interaction. Okay. But still, how could protein recognize this sequence when it's paired within a double helix?
I can't think of, but I heard that most protein binds to major groove.
Yeah, that's right. So if you remember when you have a double helix of DNA, you have the major groove here and you have the minor groove here. Okay? And here you have the bases. There. So the atoms of the bases that are involved in hydrogen bonds, they cannot be used for protein recognition. So what's happening? What does it mean when a protein binds to the major groove? Or the minor groove for that matter? It's the same thing. What will the protein, if you have a transcription factor that comes here,
maybe proteins recognize the part of the protein which is not involved in hydrogen bond.
If you have a protein here, okay, and this protein recognizes this sequence, so what the protein does, reformulate here.
What? Reformulate?
Reformulate.
Proteins attached to the part of the protein which is not involved in hydrogen bond.
The protein attached the part of the protein, so
part of the DNA base.
Yes, so that's it. So even though T and A make two double bond, two hydrogen bonds with each other, and C and G make, 3 hydrogen bonds with each other, still you have atoms that are sticking out of the helix that are different whether you have a G, a C, a T, or A. You have atoms sticking out of the helix that can be used for recognition. Okay? So even though the protein will not interact by doing this kind of pairing, still many atoms are available for non-covalent binding. And this is what's happening. And the major groove, why most protein interact with the major groove? Because it is more open. The available space for interaction, it's better. there are more possibilities for interaction in the major groove than in the minor groove. Okay? And the atoms that are sticking out of the major groove, they will be different for each sequence of nucleotides that you have. Okay? Of course, the phosphate backbone will be the same, but each base will have different atoms and present these different atoms for potential binding. So this is what happened. And if the protein has amino acids that interact with a specific sequence, and let's say this protein has a front and a back, so the amino acids here, they are well positioned to interact with the sequence here, let's say GGCTGA here, so the protein cannot bind in the other direction because the interaction will be less a good match. Okay? Maybe it could, but it will not do anything because the interaction is not so strong. And the protein will only be functional once it really finds a good match. So then it's easy to understand why the protein doesn't bind in the other direction. Okay?
The part of the base which attached to protein is unique to A and G and C and D.
Yeah, exactly. It's unique to ATCG, but it's also unique for each sequence of ACTG. The atoms available for binding in the major groove, if you have ACTG or if you have GGGG, this is completely different, and that the protein will recognize. Okay? So this guy.
Another question.
Yeah, of course.
In the case of DNA replication
Yeah.
or transcription, protein unwinds double helix to
That's right. Yeah.
single strand,
Yeah.
and I understand that AT pair is more easy to unwind,
Yeah.
but how does the protein unwind the double helix?
That's a good question. That is a good question. I really have no idea exactly of the molecular interactions that are needed, but you certainly require energy. Okay, you certainly require energy. Well, I can imagine amino acids within the protein will actually firmly bind to each of the single strand of the DNA, and probably via changes of the protein structure, they will actually attract the single strand of the DNA. So if you think that the binding of a protein to each of the single strand is stronger than the energy that keeps the single strand together, then the two strands will be pulled apart because the protein exerts a tension that is higher than the energy that keeps them together. And I can imagine that for each different protein, the mechanism of how it does it and the amino acids that are involved in interactions with each of the single strands will be different.
It's not chemical reaction because it's hydrogen but not covalent bond.
it's not covalent bond, but everything is a chemical reaction. So if you require energy, certainly you require energy to pull the strands apart. So requiring energy means using ATP, which is a chemical reaction.
Just the protein DNA reaction 親和性 it is easy to attract protein and DNA is stronger than DNA hydrogen bond. Isn't the is spontaneous, but
What do you want to say? So it's a little bit. The image is a little bit like this protein that we saw that cuts a polysaccharide chain that is an antibacterial protein. Do you remember that has this polysaccharide and it sort of bites a chain of polysaccharides? And when it bites that chain, the interaction that the atoms of that protein do with the chain of polysaccharide actually bends the polysaccharide chain in such a way that a reaction is possible. This is a little bit similar. When the protein binds DNA, it will change the structure of DNA and make it easier to separate the two strands. Exactly what atoms are involved in separating, I don't even think these things are known fully, because it really again depends on each protein interacting with DNA. And the proteins that separate the two strands during DNA replication, and the protein that separates the two strands during RNA and during transcription, that would be different. That would be different as
well. I didn't remember the polysaccharide cut.
This is very similar. It's an antibacterial enzyme that is in your saliva, in many biological fluids, that actually cuts the cell wall of bacteria and actually allow the lysis of bacteria. So I think it's chapter 4. Chapter 5. Probably not. No, this is a good thing. You know, you have a lot of, in the major groove, you have a lot of atoms there that are available for binding. So these are very important for recognizing the sequence of DNA. So this is not this chapter 4 that one. Yeah, that's it. RNA. So this lysozyme thing, this is very briefly how it does. So that's the polysaccharide chain, which is very similar to the molecule of the cell wall of a bacteria, and this lysozyme actually binds this chain and cleaves it. Okay, so actually this destroys the bacteria wall. But this is very similar to how a protein can interact with DNA and open up the two chains. Of course, reactions are different, but the key, as you can see here, so normally this is the substrate here, and the structure of a polysaccharide chain, each atom have a specific relationship with each other, and the angle between the chemical bonds is fixed, right? But once actually this polysaccharide chain binds into the active side of an enzyme, then you can see that the configuration of each atom is actually changing a little bit. The chain is bent. And because the polysaccharide chain is bent, this makes it easier to actually do chemical reactions between each of the atoms. Like you have an attack here of the oxygen with the carbon here, and you have a nucleophilic attack here as well. So, these kind of things are very important. So how a protein interacts with DNA is very similar. So all the atoms of the protein will actually pull apart the two chains by really changing the chain of RNA. And if you remember, RNA or DNA also, if you remember the last chapter we saw, there was something similar. I think that was, the last chapter we saw was 6 or 7? 7? This is 7. This is DNA replication. Even at the beginning, so that's the Tata box binding protein. When it binds to the DNA, it really changes the structure of the DNA. So the whole double helix is really kind of bent, which makes it very easier to actually separate the two strands. So this is very important. If this does not happen, it's not just putting it like this. It has to completely change the double helix and make sure that the atoms are available for acting. Exactly how it happens, I don't know. It depends on each protein. Another question? No? I'm trying to find if I can ask a question to you. Yeah, so there is on the page 256 of the book, there is a question 7.6. Discuss the following. During the evolution of life on Earth, RNA lost its glorious position as the first self-replicating catalyst. Its role now is as a mere messenger in the information flow from DNA to protein. Discuss. It's a very open question. Discuss. So I repeat again. Discuss the following. During the evolution of life on Earth, RNA lost its glorious position as the first self-replicating catalyst. Its role now is as a mere messenger in the information flow from DNA to protein.
It's not a mere messenger because many RNA is used as enzyme, such as ribosome or I'm not sure.
Such as ribosome and what else? Just as ribosomes
And before DNA and proteins, like everything was like RNAs, right?
yeah. Well, scientists seem to think so.
Yeah.
Maybe.
So like, it could have been like, I don't know.
Yeah, it could have been just everything.
Everything could have made the same stuff like.
But the I think the part of the question that is I think the questionable part of the question is as a mere messenger. As a mere messenger means that it's only messenger RNA. But there are many RNA who still have enzymatic function, catalytic function, like what you said, ribosomal RNA. But there are other things, not only ribosomal RNA. What other RNA have a kind of catalytic function? Messenger RNA, we may find in the future that some of them have catalytic function. We don't know, we think that there is no, but there are other RNA that have a catalytic function. Not only ribosomal RNA. Like what? That exist naturally, not that have been made in laboratory, that exist naturally in cells. Do you remember? It's one of the last lectures. You remember what else? Not only ribosomal RNA. What about tRNA? Do they have a catalytic function? tRNA. Do tRNA have a catalytic function? Do they have a catalytic function? For example, do tRNA catalyze the attachment of an amino acid on themselves? Do they catalyze this reaction? So you have a tRNA that says, oh, that's my amino acid. I'm going to catalyze its attachment to myself. Its attachment to myself. Is this what tRNA do?
I think no, it's the protein's role.
Yeah. So you have a protein that will attach the amino acid to the tRNA. Yeah. So will the tRNA catalyze the elongation of the peptide chain during translation? Do they catalyze that? Do tRNA catalyze the elongation, protein elongation?
It may be another protein's function.
Yeah, it's the ribosome. It's the ribosome who does it. Okay, the tRNA is just like an adapter. So what other RNA have catalytic functions among all the RNA that we saw in the cell?
Maybe RNA polymerase have RNA subunit.
No. RNA polymerase, it's a bona fide protein I think of one in eukaryotic cells for a very specific process that does not exist in bacteria.
Telomeres?
Not telomeres. It's a good guess though. Not telomeres.
This is protein elongated.
It's the telomere. It's right that the telomerase have an RNA primer to actually direct where they have to elongate in the DNA. But the RNA doesn't have any catalytic function by itself.
RNA interference.
RNA interference, that's a good guess as well, but that's not correct. RNA interference, so the RNA will, if you have a small RNA that is complementary to a messenger RNA, it may bind to it, form a local double helix that will be recognized by protein to destroy that messenger RNA. So it's a way to fight against viruses. Okay? But the RNA interference, the RNA will not have any catalytic function.
Your house brain has melted.
What?
Brain has melted already.
Yeah, it's the end of the year. Yeah, I also feel a little bit. It's the end of the year. So what else? What other RNA with catalytic function? Ribosome. Yeah, okay. We know ribosome, but what else? As the song says, I'm gonna wait until the midnight hours until you tell me what's the RNA in question. Cold light. Which RNA in eukaryotic cells have catalytic function? And what process do they? Mediate. Ah. Somebody is in the right location. Right chapter. So, so what do you think? Which RNA have catalytic function?
That's something.
Be careful because he's looking at your book. He may answer before you. Yeah, snRNA. Yeah. What does it mean, snRNA?
Almost like exons.
Small.
Small nuclear RNA.
Yeah, small nuclear RNA. What do they do, small nuclear RNA? Are they just going around, just being small? What do they do, small nuclear RNA? Small nuclear RNA.
It's something to do with exons.
Yeah, yeah.
What was the other exon and
I don't know.
exon? What was? Yeah.
Something to do with exon and something to do with small nuclear RNA. So what do they do that is linked to exon? These little small nuclear RNA, snRNAs. Yeah, that.
Did they remove exon or?
Nah.
No, exon.
Well, it's always which point of view. If you are one, you think that the other one is removed, and if
exon is connected.
exons are connected. Yeah. What is removed? What is removed? When you connect exon together, what do you remove? 何を取る?
Introns.
Introns, yes. So the removal of introns to attach exon together and make the major messenger RNA. What is this? How is it called? In French, it's called épissage, but it will not help you. How is it called? The removal of introns and the end-to-end attachments of appropriate exon.
Splicing.
Splicing, yes, splicing. And the small nuclear RNAs are the molecules that actually catalytically remove these introns. They associate with protein to make complexes, but the catalytic activity comes from the nuclear RNAs, the small nuclear RNAs. So ribosomes and these snRNAs are two examples of naturally occurring ribozymes. Okay, so RNA with catalytic activities. So no, it is not as a mere messenger. There are many other functions that are the functions of RNA.
How do small nuclear RNAs and catalytic activity? How small nuclear RNA do splicing?
It's in chapter. It's the chapter we saw. It's actually one nucleotide in the snRNA attacks a specific nucleotide within an intron at the exon-intron junction. And they make a link which actually liberates the link between the exon and the intron.
Intron junction. Does intron junction have specific sequence?
That's right, yeah, yeah.
It's universal.
You have to be very careful with words like universal, because depending on which animals you are in, the sequence can be slightly different. Even between man and human, it can be different. But yeah, it is mediated by sequence. So that's in the last chapter. In the last chapter we saw here. So now you have this is the donor exon here, and that's the intronic sequence, and this is the acceptor site of the next exon. And at the end, when splicing is finished, then this nucleotide is attached to this one. But this doesn't really happen by itself. This is where you need small nuclear RNAs complexed with proteins. So they will first attack this nucleotide here. I think, yeah, so you have this thing here, and it's all mediated by small nuclear RNAs, where you have this branch point adenine with its OH, which is used, mediated by small nuclear RNAs, to actually break this bond here. So you have an intermediate that looks like a lariat, like that. And then after that, this OH is then used to attack the acceptor site in the next exon to actually finalize the splicing. And this is mediated by a catalytic RNA, which is one of the SNRNAs. Like this. So it's wrong to think that without SNRNAs, these things can happen spontaneously. They will not. Okay. Or maybe they could, but the likelihood of it happening is so low that they will not actually. And for that, you actually need these small nuclear RNAs that will actually change the structure of the intron exon and actually mediate this reaction. So this is why it's a catalytic inactive RNA. Okay. So, by the way, what is catalysis? How can you explain what is catalysis?
Okay.
What is catalysis? How can you define what is? When you say RNA catalyzes something, what does it do? That's chapter 1 or 2. What does it do? Like, is a match a catalyzer? If I want to burn my book after the exam, and if I crack a match and use the match to burn my book, is the match a catalyst? Mm-hmm. Or a blowtorch for that. Is it a catalyst? Is it a catalyst?
Catalyst
Mm-hmm.
helps the reaction,
Yes, helps the reaction, yes.
but they are not use the reaction.
They are not used, they help the reaction, but they are not used. They can be regenerated, this is what you mean. But what does it mean, help a reaction? What does it mean, helping a reaction?
They reduce the reaction energy.
Ah, めちゃ近い. It's very, very, yeah, the なんとか energy, but not the reaction energy, the 最初の energy, なんか、どう in English, the yeah, the little bump in the energy of the molecule, right? Yeah, activation, the activation energy, yeah. So this is what they do, okay? So these SNRNAs, this is what they do. They decrease considerably the activation energy of this reaction here, right? By actually bringing the branch point adenine close to this donor side of the exon here. So then it's a catalyst. Any other questions?
does RNA except the messenger RNA go through splicing?
That's a good question. So you mean any other RNA, maybe also, could other RNA be spliced? Yeah, exactly. So ribosomal RNA are also spliced. Ribosomal RNA are also spliced, and long non-coding RNA, they are not used for protein, but they are also spliced. So splicing is not something that is specific to messenger RNA. Other RNA are spliced. Actually, the ribosomal gene has introns as well that need to be spliced before making the final ribosomal RNA. And long non-coding RNA as well, they have introns, so before they can be functional, introns must be removed, and then the long non-coding RNA can be used. So that's a good question. What is the difference between a polycistronic messenger RNA and an eukaryotic pre-messenger RNA that requires splicing?
What is polycistronic?
I think I said it once. Polycistronic messenger RNA. This is in bacteria. This is in bacteria. And I think there is one. It's in the book. The word is in the book. I'm not inventing it. Okay, that's a pre-mRNA, right, with intron and exon. And what's the difference between that and a polycistronic messenger RNA that is maybe in the previous chapter? No, one before that. Anybody has an idea before I show? What is a polycistronic RNA? Oh, that was this one. There is one figure in the book about it. Anybody has an idea what is a polycistronic RNA? Korea. That's a polycistronic RNA. That's a polycistronic messenger RNA. So what's the difference between that and a pre-messenger RNA that requires splicing? This is what you have usually in bacteria. And a gene that codes for a polycistronic RNA is called an operon, usually. Like the operon lactose, which we have seen in the beginning of this lecture, which is used by bacteria to use lactose as a source of energy instead of glucose. And actually, one messenger RNA contains many sequences that are used to code for different proteins that are all required for using lactose as an energy source. So what's the difference between that and a spliced pre-RNA?
Eukaryotic pre-mRNA does not code more than one protein.
That's very tricky, isn't it? Because actually, if you have alternative splicing, one pre-messenger RNA could code potentially for many different types of protein.
At the same time.
At the same time, yes. That's at the same time. In this case, for a polycistronic RNA, you have one messenger RNA, and at the same time, many proteins are made, because as you can see here, there are 3 initiation codons, and in bacteria, ribosomes can actually directly start translation in the middle of the messenger RNA. Usually in eukaryotes, then the ribosomes needs to go in the 5' cap and then zip along until it finds a start codon. But in this case, they can really bind in the middle until they find AUG and start. So this is different. Okay. Simultaneously, as polycistronic messenger RNA can code for many different proteins. Which means that the polycistronic RNA has many different ribosomal start sites. Okay, this is also one potential answer. There is more than one start codon, although this can also sometimes be the case for messenger RNA in eukaryotes. So it requires quite a long explanation to actually tell the difference. And what's the advantage of having polycistronic RNA? So if you're a bacteria, what's the advantage?
Time.
Yeah, time. Why time?
Well, doesn't it splicing, and you can like make several proteins at the same time.
Yeah. So it's save time and save energy. And if you require something that has many steps, like using lactose for energy, then you need to change lactose into many intermediates that each require different enzymes. So instead of having many different genes that code for different proteins, you have one gene that codes for all the enzymes you need. So this is like a set. When you go to the shop and you buy a set of something instead of just one tool, that's the same thing here. Okay. Instead of just buying one pen for one purpose, you buy a whole box with many pens for different purposes. That's the same thing. Okay, any other question? I'm the one asking questions, so you should ask questions.
So why eukaryotes doesn't have operons or polycistronic messenger RNA?
Yeah, that's a good question. Yeah, because it's also important for eukaryotic cell to save energy, right? So it's a very good question. Why they do, we have no idea. Why they do, we have absolutely no idea. You can say they are more complex and they are regulating things in a more complex way, but that's not really an explanation. It's just a guess as to why. But
I think it's more flexible
Yes, in this case, once they are tied, then that's it. But since the behavior of eukaryotic cells is more complex, then it needs to be regulated in more complex ways. And also, why we are having introns and when introns evolved, we also have no idea. There are two theories. The one theory is that introns are very old, but they disappeared during the development of bacteria. And the other theory is that introns is a new invention for eukaryotic cells, which is correct, we have no idea. So introns are very old and they disappeared in bacteria, or introns are very new and bacteria never had, and it's a kind of eukaryotic invention. So we have no idea. We have no idea. All right, so if there is no question, you are free to go. Okay, I'm not going to say anything new today, so you are free to go at the time.